go ahead and get started with the introduction, and we'll let people join in by the time Dr. Scaglione starts. So good morning, and thank you everyone for joining us for our very first Science Showcase. My name is Lauren Moore, and I'm the Chief Scientific Officer at the National Ataxia Foundation. So thanks to our generous donors, the NAF is able to fund about $800,000 in research grants each year. And so this webinar series is going to give our funded researchers a chance to highlight the advancements they've made in research as a result of some of that seed money grants. So we hope that these talks will encourage scientific discussion amongst researchers and to help educate our donors on how research funding is being used to advance our understanding and treatment of ataxias. So before I introduce our first speaker, I need to run through just a few housekeeping items. So first, you can turn on closed captioning by clicking the CC icon in your Zoom menu. And throughout the talk, you are also free to chat to other attendees using the Zoom chat feature. We strongly encourage and have set time aside at the end of the talk for questions. And so you can submit questions throughout the talk using the Q&A feature, or during the live Q&A session at the end of the talk, you can also use Zoom's raise hand feature to request to be unmuted and to ask your question live. And so with that, I'd like to introduce our first speaker, Dr. Matt Scaglione. Dr. Scaglione is an assistant professor in the Department of Molecular Genetics and Microbiology and Cell Biology at Duke University. The Scaglione lab focuses on two main projects. The first focuses on an amoeba that is naturally resistant to polyglutamine aggregation. And the second focus of the lab is on how mutations in the neuroprotective E3 ubiquitin ligase chip cause SCA-48. So together, the goal of the Scaglione lab is to understand basic cellular pathways that cause or prevent neurodegeneration and to translate these findings into therapeutics. So today, Dr. Scaglione will present on findings from a research seed money grant that he was awarded in 2022, focusing on mechanisms of neurodegeneration. And so with that, the floor is yours, Dr. Scaglione.
All right. See if I can share my screen here. Thank you, Lauren, and everyone at NAF for setting this up. And I'm excited to tell everyone a little bit about some of the work that has been funded by NAF in my group. So just check in real quick. Does everything look okay? All right, great. All right. So my lab is generally interested in neurodegenerative diseases, and specifically, we study a number of ataxias. So there's 4 individuals up here you may be familiar with. So this is Ronald Reagan, who succumbed to Alzheimer's disease. And if you're a baseball fan, you'll know Lou Gehrig here, who suffered from ALS, or Lou Gehrig's disease. Third, we have Muhammad Ali, who suffered from Parkinson's disease. And this last one's Woody Guthrie, who passed away from Huntington's disease. And while all 4 of these individuals had different symptoms and can be differentiated in the clinic, one commonality of these people is they all suffered from neurodegenerative diseases that are characterized by protein aggregation. So that's really the underlying goal of my lab, is to understand how protein aggregation contributes to neurodegenerative diseases and try to identify if we can develop therapies based on our basic science findings. And as Lauren mentioned, there's really 2 main projects in the lab. One project which focuses on an amoeba, Dictysphaelium discoideum, which I'm not going to talk about today. And the second project, which has really been growing rapidly in the lab, is focusing on interrogation of a neuroprotective E3 ligase called CHIP that has, in the past decade, been identified as the cause of 2 neurodegenerative diseases, Spinocerebellar ataxia autosomal recessive type 16, or SCA-16, and SCA-48. So as I mentioned, my lab is interested in protein aggregation in neurodegenerative diseases, and there are these different diseases that can be caused through different mechanisms, whether that be sporadic diseases like Parkinson's and Alzheimer's, which are predominantly sporadic, or inherited diseases such as the Huntington's disease, where a polyglutamine tract becomes expanded in individuals and is passed down from generation to generation. Clinically, these are distinct diseases resulting in different things such as dementia and Alzheimer's disease, or movement disorders and Parkinson's. And that's because they affect different regions of the brain. And this is caused by different proteins, such as the amyloid beta protein, which causes the characteristic A-beta plaques, which accumulate in the Alzheimer's disease, or accumulation of proteins like alpha-synuclein, which forms these cytosolic Lewy bodies in Parkinson's disease. Or again, intranuclear Huntington, which accumulates in the nucleus of cells in Huntington's disease. And while all these diseases are very different in terms of the presentation of these aggregates, at a molecular level, they're actually all formed by the same, are all caused by the same formation of amyloid. And so really understanding how cells counteract the accumulation of this fold of amyloid is one main focus of the lab. And cells really have a few different ways to do this. So in cells, there's around 20,000 different proteins which adopt native folds, and those proteins then have a job to do within the cell. And whether that's due to environmental conditions, such as cellular stress or genetic mutations, what can happen is these proteins can tend to misfold, in which case they adopt an improper conformation and can aggregate. And the cell has developed a number of pathways to help resist this process of protein misfolding. So it has pathways such as the ubiquitin proteasome and molecular chaperones. Molecular chaperones can help to assist in turning these proteins back into the properly folded form, while things like the ubiquitin proteasome system basically serve to target these proteins for degradation, so just to remove them from the cell. Now, if these pathways fail, you're going to have an accumulation of some sort of proteotoxic species, and this proteotoxic species is going to drive cellular death. However, if these pathways are successful, you suppress the accumulation of this proteotoxic species, and this can promote cell survival. And this is extremely important in neurons because these are postmitotic cells which are not actively replicating. So promoting cell survival is really one of the ways we think that we can use to treat neurodegenerative diseases, is to prevent the neurons from degenerating in the first place. And I really want to go a little bit deeper into one of these pathways, which is this ubiquitin proteasome system. So this is, again, one of the ways that which cells can target select proteins to be degraded in the cell. And ubiquitin itself is a small 8.6 kilodalton protein. So it is activated by one of two E1 ubiquitin activating enzymes in an ATP-dependent manner, where it forms a thioester bond in an active site cysteine residue. So you can think of this as the first step of turning on this pathway. E1's job is to take ubiquitin, activate it, and then hand it off to something called an E2 ubiquitin conjugating enzyme, of which there are roughly 40 different E2s in human cells. Select E2s can interact with E3 ubiquitin ligases. So E3 ubiquitin ligases can be thought of the scaffold, which is bringing the E2 ubiquitin conjugating enzyme and the substrate into close proximity to facilitate the transfer of ubiquitin from the E2 to typically a lysine residue on the substrate. And E3s are actually a very large class of enzymes. There's thought to be roughly 600 different E3 ubiquitin ligases in human cells. And then finally, this process is reversible. There's something called deubiquitinating enzymes. Proteins have been well studied. You may be familiar with one of them, which is called Ataxin-3, which is the polyglutamine protein that becomes expanded in SCA3. And so deubiquitinating enzymes are roughly nearly 100 in human cells. And through the coordinated function of the system, then, proteins can have chains of ubiquitin attached to them, and these chains serve as signaling functions. So you can think of this as kind of like a postage stamp that sends a protein along to its destination. That can be the proteasome for degradation, or alternatively, it can alter protein-protein complexes or affect functions of the proteins within the cells. So due to the fact that roughly 5% of the human genome encodes this system, we believe it's a really powerful system in order to potentially develop drugs for it to treat diseases, including neurodegenerative diseases. And so my lab has a large focus on one particular E3 ubiquitin ligase. Its name is CHIP, and we've decided to study CHIP because it sits at a really unique place amongst protein quality control pathways. So it has an N-terminal domain called a TPR domain, and TPR domains function to recruit molecular chaperones like HSP70. Chaperones bound to misfolded proteins, and CHIP can also bind those E2 ubiquitin conjugating enzymes I mentioned earlier. And that facilitates the transfer of ubiquitin from these E2 ubiquitin conjugating enzymes to the misfolded proteins that are associated with chaperones. And ultimately what that will do to lead to their degradation via the proteasome. So overall, CHIP really sits at a critical juncture between protein folding and protein degradation. And understanding how CHIP functions in cells, we believe is a really important way to develop therapies for neurodegenerative diseases caused by protein aggregation. And so this is just kind of a schematic here showing you the crystal structure of CHIP. We'll go more into this in a bit. It's an asymmetrical homodimer, so there's two CHIP molecules here. CHIP interacts with the C-terminus of chaperones like HSP70, which again bind to these misfolded proteins to facilitate their ubiquitination, ultimately targeting them to the proteasome for the degradation. So this system's been known for about 25 years. CHIP's been fairly well studied, and we have a pretty good understanding of this particular aspect of CHIP function. In vivo, CHIP's a very important protein. So one of the features of neurodegenerative diseases, they're age-related, and deleting CHIP in mouse models leads to an early aging phenotype. So up here, you can see a wild-type mouse at 3 months of age versus a CHIP knockout mouse. You'll notice the kyphosis, so this curved curvature of the spine here, and that they're slightly smaller in stature at 3 months of age. And by 12 months of age, this becomes very pronounced. So you can see here's a 12-month-old wild-type mouse versus a 12-month-old CHIP knockout mouse. And this CHIP knockout mouse looks closer to a 24-month-year-old, which would be an old mouse. So there's this accelerated aging phenotype. This is reflected in their lifespan. So if you can see in this particular study, the point at which 50% of the wild-type mice had died is around 25 months of age. However, in CHIP knockout mice, by 7 to 8 months of age, half of the mice have passed away. So this tells you that CHIP really is a protective protein that protects against aging. And importantly, it's also protective against cellular stress. So this is an experiment where they took a mouse and heated it up to 42 degrees Celsius. So heat stress is commonly used as a general stressor. And what you can see is that about 75 to 80% of the mice, wild-type mice, survived. However, CHIP is absolutely essential for viability following heat stress in mice. So within 7 hours post-heat stress, 100% of the CHIP knockout mice have died, informing us that CHIP's really a key protective protein against cellular stress. And this shows up in disease as well. So this is work done from Hank Paulson's lab looking at a mouse model of Huntington's disease with a single copy of CHIP deleted. So we have our wild-type mice here, mice with a single copy of CHIP, a mouse model of Huntington's, and then a mouse model of Huntington's with only a single copy of CHIP. So you can see in this particular panel that there are behavioral abnormalities that come with deletion of one copy of CHIP. And importantly, there are losses in viability as well. So again, we have our wild-type mice, our CHIP haploinsufficient mice up here. If we have a mouse model of Huntington's disease, those mice begin to die around 10 weeks of age, and you can see that this happens much more rapidly when there's only one copy of CHIP. So much like resisting cellular stress, of a general cellular stress like heat stress, CHIP also protects against things like neurodegenerative diseases in vivo. Now, more recently, our labs have become interested in CHIP because mutations in CHIP have actually been shown to cause neurodegeneration. So CHIP's encoded by a gene called STUB1. So if you see STUB1, that just refers to the gene that encodes the protein CHIP. And so you can see kind of a cartoon schematic of the domains of CHIP. Again, it has a TPR domain. So this is the region that recruits molecular chaperones. It has a coiled domain, which mediates its dimerization, shown here in green. And it has a U-box domain, which recruits those E2 ubiquitin conjugating enzymes. And starting in about 2014, there were identification of recessive mutations that cause SCA-16. I'm only going to briefly speak about these. We've done some work on these. These appear to be loss of function mutations, which destabilize CHIP, and I'll show you a little bit of that data. But more recently, what we've become interested in is mutations that cause SCA-48. So SCA-48 is a dominant disease, or what I'll tell you about a little today, potentially digenic, meaning that there's a second gene that's mutated in many of the patients which have SCA-48. We've characterized many of these mutations to show a loss of function of either chaperone binding or E3 ligase activity. And while I won't talk about it today, there's some potential gain of function as well. And I just want to kind of spend some time looking at the structure of CHIP here. The reason that we became interested in SCA-48 was that there were clear regions of the protein where mutations accumulated. So the domains are colored in a manner that corresponds with this diagram up here. And the mutations that cause SCA-48 are shown in red. And it's up to about 50-ish mutations which have been identified. So that's roughly 1 in every 6 amino acids in CHIP can be mutated to result in SCA-48. But what you'll see is that these aren't just randomly distributed around the protein. They map to specific protein-protein interfaces within CHIP. So for example, in the TPR domain, which the mutations I'll talk about today, they really map between this first and second TPR repeat. So almost every single amino acid that maps kind of where these two helices pack up against each other is mutated in SCA-48. We also see that they map to domains where U-box and the TPR domain bind to each other, as well as to regions which mediate interactions which facilitate dimerization. So we thought that was interesting because with SCA-16, the mutations just seem to sporadically map all over the surface of CHIP with really no clustering like we're seeing with SCA-48. So back to SCA-16. So we want to start with telling you a little bit about SCA-16, because this is really what informed us to ask the questions that we're currently asking about SCA48 and chip function. So again, here's a list of the mutations which cause SCA-16, and here's an in vitro ubiquitination assay. So I mentioned that chip's a ubiquitin ligase. So here's a substrate. So in this case, we use HSP70 as a model substrate. And if we put chip and all the enzymes that facilitate the attachment of ubiquitin in, what you can see is you get this slower migrating species during SDS page and a Western blot, and you get this laddering effect. So each of these little lines that you're seeing is the attachment of a ubiquitin to HSP70, resulting in polyubiquitinated HSP70. And if you look at the SCA-16 mutants, what you can appreciate is that you have a loss of this higher molecular weight species, so a loss of the stuff that's occurring up here at top, and accumulation of unmodified mono or diubiquitinated chip, suggesting that this SCA-16 mutants decrease the ability of chip to ubiquitinate their substrates. I should mention these reactions are all done at 37 degrees Celsius. These are human proteins, and that's the physiological temperature. And so we were wanting to really understand how these mutations were affecting chip function. So we did a series of experiments, but the one that I thought was really telling is we did a simple experiment where we looked at the unfolding of proteins. So this is an experiment where you put a dye in a test tube with your protein of interest, and then you simply heat that mixture up 1 degree Celsius at a time, from 4 degrees all the way up to 95 degrees Celsius. And as the protein unfolds, the dye will stick to that protein, and that makes the dye fluorescent. So you can measure the unfolding of the protein. And we were surprised when we looked at chip that we found that it had a melting temperature of 39 degrees Celsius. So this is very low for a protein. It's only 2 degrees above physiological temperature. And so that seemed kind of strange to us. And what we found was that actually, if you look at the vast majority of mutations which cause SCA-16, they're simply dropping the thermal stability of chip a few degrees to be below physiological temperature, which again, as I mentioned, is where we do all of our assays at. There are a few other mutations which have defects in HSP70 binding or loss of E3 ligase activity. But for the vast majority of them, it seems to be simply thermally destabilizing the protein, so it's not able to properly fold at 37 degrees Celsius. So we came up with the idea that if this is really the case of why these proteins are losing activity, that we should simply be able to lower the temperature of our test tube when we do our reactions, and we should be able to restore activity. And that's exactly what we saw. So again, this is a ubiquitination reaction, just like I showed you before, except this time we did the exact same reaction at 25 degrees Celsius. And I think what you can appreciate is that there's a much more of this high molecular weight ubiquitination in most of the samples, and much less of this unmodified accumulating. They look much more like the wild type protein. So what that told us was that these SCA-16 mutations basically are thermally destabilized in such a way that causes them to be unable to bind to and ubiquitinate substrates. Okay, so that's a little bit of background. Fast forward a couple years later, and I received an email. Oh, sorry, one more slide. An email from Pedro Gonzalez, who's a neurologist, who had a patient who had been identified with a mutation, in this case, a dominant mutation, this A52G mutation, that they thought may be the cause of this patient's ataxia. And he asked us if we could take a look at this protein to look to see if it may be causative of SCA48. I'm not going to go into all the data from that. It maps to that same interface in the TPR domain that sits between the first and second TPR repeat. But this is one of the experiments that really kind of has guided us moving forward, that was done in collaboration with Rick Page's group. So this is a CD spectroscopy plot. And the important part to note is that you see these dips down around 208 and 216 nanometers. And these correlate with an alpha helical fold. So this is a way to look at the structure of chip at different temperatures. So Rick's lab went through and basically, again, raised the temperature of chip 1 degree at a time, from 4 degrees all the way up to 95 degrees, and asked, how did this change? And what he found was really interesting. So this is a different way, again, to look at unfolding of chip. In black, we have wild type chip here. So you can see that there's an unfolding event, which happens at 39 degrees. And then there's a second unfolding event that happens with a TM of around 56 degrees. So that's telling us what we were monitoring before was this first unfolding transition with the SCA-16 mutants. So there's this 2-domain unfolding event. What he also found was that if you look at this A52G mutant that causes SCA48, you can see that there's less helical content to begin with. So you can see the shift between these two lines. But perhaps most interesting, what he found was that this loss in intensity is only in this first unfolding event. So you can see that it comes together after that first unfolding is done, and then it progresses to unfold at the same time. So whatever this mutation is doing, it's affecting this first unfolding event, which is happening at very low temperatures. And so this mutation falls in the TPR domain. So this led us to hypothesize that this first unfolding event was something happening with the TPR domain of CHIP. And so we went through and we really took a look at the mutations. It's really quite striking. We won't even further down in plants now, but the mutations that cause SCA48 are highly, highly conserved within CHIP. So we've been down, I think the last organism we looked at was something called a sea squirt, which also had nearly 100% conservation. There was only one amino acid that was slightly different. And so in red here are the mutations which cause SCA48, and you can see that they're all identical from plants all the way up to humans. And again, they map this interface between this first and the second TPR repeat. And so we wanted to look at this structurally. So we turned to using a technique where we add a protease called trypsin, and trypsin will then degrade recombinant proteins. So it'll cut it up into smaller peptides. So this is unmodified CHIP here. And if we put trypsin in, you can see that it gets digested here with an accumulation of this slightly smaller species pretty rapidly. And if we look at SCA48 mutations, so this is again the A52 gene mutation, you can see that this band accumulates more rapidly, suggesting that this is less structured and that we're losing this region of the protein first. So we isolated this band and sent them off for mass spectrometry to ask what part of the protein is being digested here. What are we losing? And what we identified, as expected, was that this first TPR repeat is being lost. So up to amino acids 51, peptides that are associated with that region are heavily missing from this structure. And so if we kind of map that out, in red here again are mutations which cause SCA48, and kind of partially transparent here is the region of CHIP that we're not detecting by mass spectrometry. So this region's more unstructured and more liable to degradation by trypsin. We also use protein NMR to directly look at the TPR domain. So in this particular plot, we have in black is wild type CHIP, and in red is SCA48 mutant CHIP. So this is again the A52 gene mutation. And you can think of each one of these little peaks or dots as you're seeing here as a single amino acid. So this is a coordinates which map to specific amino acids. And we simply just took a look at wild type and SCA48 mutant CHIP. And what we found was that there was a loss of intensity of peaks that again correlate with this first TPR repeat. Again, confirming our data from our partial trypsin digest data that this region is losing structure. Moreover, if we simply heat the protein up in a test tube, and there's a lot of controls, and I'm not going to go into all the details here, but we essentially see the same thing. So whatever structural chain that's happening with SCA40 mutations, we can mimic by simply warming the protein up past its TM, up to 45 degrees Celsius. And that's kind of shown what we had by NMR here. Again, we have this region that goes, that we have a loss of signal for by NMR. There's additional residues that are lost here. This is due to technical reasons, as this isn't packing up against them anymore. So their spectrum changes as well. But the take-home message from this is that these SCA40 mutants induce structural changes to the TPR domain of CHIP that also seem to occur when we slightly heat CHIP up. Now, the prediction from this is that it won't bind chaperones anymore. I'm not going to show you all the biochemical and biophysical data. We've done a number of series exposing that. But what I want to show you here is that this happens in cells as well. So this particular experiment is a split nanoluciferase experiment. So we have half of luciferase attached to CHIP and half attached to HSP70. And so you can see here that if we have the constructs at 37 degrees in cells, we can see the interaction between CHIP and HSP70 compared to a negative control. And if we simply heat these cells up to 42 degrees Celsius for an hour and come back and measure them, we can see that there's roughly a 40 to 50% loss in signal, suggesting that CHIP and 70 are coming apart in complex. Moreover, if we do the same assay, but now look just at 37 degrees and look at 8 different mutations which cause SCA48, all located within that TPR domain, you can see that there's a dramatic loss of interaction with CHIP and HSP70 between these proteins in cells. Again, consistent with the idea that there's structural changes in SCA48 that are causing difficulty with CHIP binding to molecular chaperones. In addition, we also did assays to look at the ubiquitin ligase activity of CHIP with SCA40 mutations and with heat stress. So this is called a thioester discharge assay. These are, again, recombinant proteins in a test tube. And we take the E2 ubiquitin conjugating enzyme, UBCH5, which is shown here, and we add in all the things that are necessary to charge its active site with ubiquitin. So that's a slower migrating band here. So negative control, you can see it's missing. And then we simply add free lysine, which then can attack that thioester bond to cause the E2 to discharge ubiquitin. And so you can see that there's some discharge occurring just with excess lysine, and that this is accelerated by CHIP or SCA4 mutant CHIP. So these TPR mutants selectively cause a failure to interact with things like molecular chaperones, but they don't appear to affect its E3 ubiquitin ligase activity. Similar things can be seen with heat stress, where again, we have this charging assay. If we add in free lysine, you can see that if CHIP's present at temperatures ranging from 25 to 50 degrees Celsius, it fully discharges this ubiquitin. So these assays and a number of others have informed us that CHIP basically retains its E3 ligase activity, however, it loses the ability to interact with chaperones. And so this had us thinking back into the literature about what was really known about CHIP. So CHIP's described as a predominantly cytosolic protein. So this is an image from our lab looking at a mouse embryonic fibroblast expressing a GFP or green tagged CHIP. And you can see that this place where it's largely excluded is the nucleus. And work from Cam Patterson and a couple other groups had shown that essentially when you heat stress cells, CHIP redistributes to be equally localized between the nucleus and the cytoplasm. And so because we saw similar structural changes to heat stress CHIP as we saw with SCA40 mutants, we hypothesized that similar localization changes may be occurring within cells. So we went on to make stable cell lines that are expressing either CHIP, GFP tagged CHIP shown up here, or SCA48 mutant CHIP, and then looked to see if we saw exclusion of CHIP from the nucleus. And similar to heat stress, which you can see over here with this G33S mutant is, while you can see CHIP excluded in the wild type, in the mutant, you see that it's evenly distributed through the cells. And so we did this with all 8 mutations, blinded it, and had someone in the lab analyze it. And what we found that there's a very striking change in localization from CHIP. So if we looked for the percent of cells in which CHIP was excluded from the nucleus or largely excluded from the nucleus, we see that basically with these SCA40 mutants, all of them redistribute partially to the nucleus and at almost 100% of the cells. So this is a really striking phenotype and arguing that CHIP was mislocalizing to the nucleus with SCA48 mutations. So I'm going to shift gears a little bit. So when we started thinking more about nuclear CHIP, we were getting all this data. There were 2 clinical reports that came out a year ago, a little over a year ago now. And these identified a genetic link between CHIP and TDP. And this really got us thinking about, what other roles CHIP may be playing in cell, because everything I've been telling you up to now is that it interacts with chaperones and target things for degradation. So what's TBP first? So TBP is a polyglutamine protein that and it stands for TATA binding protein. So this is one of the major proteins that initiates transcription in cells. So CHIP has this, or TBP has this polyglutamine expansion. In individuals with less than 40 glutamines, there's no disease. In individuals with greater than 49 glutamines, they develop SCA-17. And what now I think we're up to 3 or 4 clinical papers now found was that in individuals who had between 41 to 48 glutamines, and so these are individuals who had an incomplete penetrance, so some of the people would develop neurodegeneration, others would not. In the ones that were developing disease, what was found is that there were also mutations in CHIP. So there seems to be a strong genetic link between CHIP and TBP. And so the question really became, how would CHIP be leading to neurodegeneration in complex or in tandem with something like TBP? As I've told you before, what we know about CHIP now is that it's involved in this kind of end stage of a protein's life cycle. So we have DNA, which gets transcribed to RNA, which becomes translated to protein. Eventually, those proteins need to be degraded. And that's what the canonical view of CHIP is, that it targets proteins that misfold for degradation. However, what this human genetic data is arguing is that there's an unappreciated role for CHIP in transcription. And so that's kind of how our mindset shifted with this nuclear CHIP being the site of transcription is in the nucleus. We were kind of interested in looking at this link. And so what we did next is to do a bioID experiment. And so what I'm going to tell you about is a novel interaction between CHIP and a protein called TF2A. So TF2A is a protein cofactor of TBP. So TBP, again, is the protein mutated in SCA17. It binds to DNA and assists in its initiation of transcription by this transcriptional initiation complex. And TF2A is a key protein that interacts with TBP to help stabilize this interaction. So we were thinking back to our data, seeing this relocalization of CHIP to the nucleus with SCA48 mutants, and we wanted to ask, what could CHIP potentially be doing in the nucleus? And so we performed what's called a bio-ID experiment. So in this experiment, we hook a biotin ligase called BRA to CHIP, or MTs used for mini turbo, it's a variant of it. And then this protein can come through and label proteins that are in close proximity to CHIP or to a control not fused to CHIP. And then we can ask what proteins are preferentially enriched with biotinylation. And so we first checked to make sure that we had things like molecular chaperones, which we know bind CHIP. So we have our HSP70s, our HSP90s. These proteins are heavily enriched in this experiment for being near CHIP. But what we also found was that there was the presence of this protein TF2A. So TF2A seemed to selectively bind CHIP over negative control, and it seemed to do so in a really significant way. So its significance and log fold change are higher than molecular chaperones, which are known interactors of CHIP. And importantly, we did the same experiment now, looking at an SCA48 mutant and comparing that to CHIP to look for things that have defects in interacting with the TPR domain of CHIP when there are SCA48 mutants. Again, we have molecular chaperones, which have decreased labeling in this particular experiment. And we also found that TF2A, again, was one of the highest loss of signal in our mass spectrometry. So really identifying an unexpected interaction between CHIP and TF2A. So we did a number of controls to confirm this. First, we looked in our bio-ID experiment to confirm that these mutations weren't just causing less CHIP to be around, and in fact, they look very similar. And this is kind of showing you the data normalized. So if we look at the number of peptides that are identified in our mass spectrometry experiment, you can see there's a pretty dramatic loss of TF2A peptides in the A52G mutant to the point that it's virtually statistically identical to our negative control of mini turbo alone. We also used our split luciferase system to look for TF2A's interaction and confirm that we could see similar interactions that we saw via bio-ID. And again, you can see that there's a pretty strong interaction between CHIP and TF2A in cells. And then finally, we came back through and used our panel of mutants again to look to see if these SCA48 mutants caused a loss of the interaction between CHIP and TF2A. And again, all of the TPR mutants that we tested, which is a little over half of them that exist, caused a decrease in the interaction between CHIP and TF2A in cells. So what do we know about how proteins bind the TPR domain of CHIP? So there's been nice work done with the crystal structure of CHIP bound to a C-terminal peptide of HSP90 in this case. So how CHIP binds to these proteins is that it interacts with 3 key residues, and this has been a lot of really nice work done from the Jason Giswicki's group, that typically terminate in VD. So there's a lysine residue in CHIP in this TPR domain that forms what's called a carboxy clamp on this last residue in the sequence of HSP70. So it binds to the C-termini of these proteins. And what's important is a hydrophobic residue here, and then either a VD or a PD motif at the very C-terminus. So this is how it kind of binds in. And what we found interesting was that when we looked at the sequence of TF2A, it's interesting in that it's a protein that gets cleaved by a protein called taspase-1. And when taspase-1 cleaves TF2A, it now ends in what looks like a very nice motif for binding to the TPR domain of CHIP, and that would fit with our data as well. So this would suggest that TF2A can compete with chaperones for binding to the TPR domain of CHIP. And we confirmed that if we mutate this VD residue on TF2A, we have a loss of this interaction. So we simply put 2 stop codons here so that we have protein expressed in cells which lacks this VD. And you can see that there's a vast reduction of this interaction. Again, consistent with the idea that TF2A is binding to the TPR domain of CHIP. We also generated rhodamine labeled peptides. So these are fluorescent peptides where we can use a technique called fluorescence polarization. So in this assay, the way to think about it is we have peptides that correspond to the last 5 amino acids of TF2A or HSP70. They have a fluorescent dye on them. And we can use an instrument in the lab to measure their rotation, so the rate at which they tumble. And when something large binds to it, that basically slows down the speed of tumbling, so you can measure this change in polarization of light. So we titrate in, so we have 20 nanomolar peptide, and we titrate in increasing amounts of CHIP. And what you get is this nice binding curve to HSP70 peptide with an affinity of around 2 micromolar. We also found that we saw a nice binding curve to TF2A, albeit with slightly lower affinity, so about 5-fold lower affinity. Importantly, we could also show that this TF2A peptide, if we had a non-fluorescent version, could actually compete with HSP70 for binding on CHIP. So in this particular experiment, what we're doing is we have HSP70 that's fluorescently labeled, pre-bound to CHIP, and then we're titrating in increasing amounts of either the C-terminal peptide that corresponds to TF2A or a scrambled peptide. And what you can see here is that in blue, in TF2A, we're able to compete HSP70 from the TPR domain of CHIP, while with our scrambled peptide, we don't see any change in signal. Again, consistent with the idea that TF2A is interacting with the TPR domain of CHIP and competing with HSP70 for this binding site. Also importantly, if we repeat this experiment with our recombinant protein that corresponds with SCA48 mutations in the TPR domain, you can see that there's a loss of affinity. So we can see nice binding by wild type CHIP here in black, and the shift to the right indicating a pretty dramatic decrease in affinity for this peptide with SCA48 mutations. CHIP's a ubiquitin ligase, so our natural hypothesis was that CHIP is ubiquitinating TF2A. So we went back to our in vitro ubiquitination assays and performed assays looking at TF2A ubiquitination in a test tube. So here we have TF2A, and you can see if we put CHIP and the ubiquitination machinery in there, we get this laddering effect, which is consistent with ubiquitin being added to TF2A. Moreover, consistent with our in-cell and in vitro binding data, we see that if we have mutations in the TPR domain that cause SCA48, we can see that there's a decrease in the ability to ubiquitinate CHIP. And we have a control down here showing that these are all active enzymes and that you get nice auto-ubiquitination of CHIP. So this is a process by which CHIP puts ubiquitin on itself. Because CHIP's been described as targeting proteins for degradation, we next wanted to look at the levels of TF2A in cells overexpressing CHIP. So these are HEK293 cells, which were made to stably overexpress CHIP. So we have 100% of the cells expressing CHIP. You can see the overexpressed CHIP here versus the endogenous levels. So it's a pretty high level of overexpression. And when we looked at endogenous levels of TF2A, what we actually found is that we don't see differences in levels. So this suggests that while CHIP's able to interact with and ubiquitinate TF2A, it's not sufficient to cause changes in its steady state levels. And this fits with a number of other experiments that we've done in the lab looking at some new CHIP substrates that we've identified where CHIP is not necessarily targeting these proteins for degradation. Instead, what we think is that CHIP is functionally influencing TF2A's role. So ubiquitination through lysine 63, for example, on ubiquitin can lead to polyubiquitin chains that serve more to reorganize things with the cell rather than target for degradation. So our data suggests that that's what's happening with TF2A. So we next wanted to look at the functional output of TF2A in cells. So our hypothesis is that it's altering TF2A function. So TF2A is a transcription factor. It's a general transcription factor. So our hypothesis was if CHIP was important for TF2A function, if we delete CHIP, we should have changes in RNA metabolism. And that's exactly what we saw. So these are in wild type versus CHIP knockout MEFs. So here you can see the STUB1 gene, which encodes for CHIP. You can see that that transcript is vastly reduced, but we get a roughly about 2,200 different RNAs that have differential expression in the absence of CHIP compared to the presence of CHIP. Moreover, physiologically, what we know is that CHIP, again, as I talked about earlier, is really important under conditions of cellular stress. So because CHIP's absolutely essential for viability following cellular stress, we hypothesized that deleting CHIP would cause changes in how the cell responds to cellular stress at the level of RNA. So we performed RNA-seq experiments post 1 hour heat stress, comparing wild type MEFs here. So this is pre and post heat stress. As expected, we see roughly 3,000 genes have differential regulation or differential levels of RNA in response to heat stress. We get your classical HSP70s, regulated Jun, DNA JB. So these are the HSF1 targets. So these are things that have been well characterized in response to heat stress. And interestingly, if we delete CHIP, what we see is a massive reduction in the response to cellular stress. So about 70% of transcripts that are differentially regulated under conditions of heat stress, this response is lost in cells that lack CHIP. Also interestingly, we saw that these HSF1 targets appear to be still differentially expressed. And this was surprising because some work done a number of years back suggested that CHIP was actually essential for HSF1 function. But our data are look a little bit different, using these little more advanced techniques that are newly available, kind of come through and show that these HSF1 RNA levels, our targets of RNA levels at least, go up. And this would also be consistent with some literature that argues that this could be TF2A dependent because HSF1 targets appear to be not relying on TF2A for their production, whereas other genes are. And so overall, if we kind of compare these two results, what we see is about 70% of all gene changes that occur in response to cellular stress are reliant on CHIP. And so we kind of wanted to take this back now, thinking about the different things that we've learned functionally about CHIP. So one of the things about heat stress is that it's a transient stress. And one of the things that we'd kind of noticed throughout our studies was that the structure of CHIP, when we had these structural defects, it wasn't resulting in the characteristic protein aggregation that would occur. Instead, these proteins behave relatively normally in a test tube as far as their solubility and everything else. So this suggested to us that potentially we may be able to reverse the structural changes that are seen with heat stress and potentially with SCA48 mutations. So this is a pretty simple experiment. We simply took N15 labeled TPR domain of CHIP in black here, and looked at it at 25 degrees Celsius in an NMR tube. So you can see all the peaks are present. It looks happy like it should. And if we turn that up again to 45 degrees, you can see in blue. It's kind of hard to see since we have other colors here. But again, there's the same loss of signal of these first two TPR repeats of CHIP. However, interestingly, if we simply took the sample, turned the temperature back down to 25 degrees Celsius, let it sit for a couple minutes, what we saw was that the structure of CHIP in green here actually goes back to looking completely like wild type CHIP. So at least in conditions of heat stress, these changes in the structure of CHIP that also appear to occur in SCA48 are reversible. Functionally, we see the same thing. So if we do an FP-based experiment in blue, or here is wild type CHIP binding to the chaperone HSP70 at 37 degrees. If we take this exact same plate and simply heat the temperature up to 42 degrees, you can see that there's a loss in binding, again, consistent with this unfolding event that occurs. And if we take that exact same plate and simply turn the temperature back to 37 degrees Celsius, you can see that there's 100% recovery in activity. So this ability to interact with chaperones is completely reversible. Moreover, its functional activity is also completely reversible. So this is the ability to ubiquitinate chaperones. This is likely due to its ability to bind chaperones. So we can see that at higher temperatures, you have an inhibited ability to interact with chaperones and ubiquitinate them. But if we simply take these samples and put them back at 37 degrees for a few minutes, you can see that there's a recovery. So loss of ubiquitination here, you can see that that's recovered here. And finally, if we look in cells, we can see the same thing. So again, monitoring the CHIP HSP70 interaction using our split luciferase system, you can see that we have this loss of binding due to heat stress. And if we simply let the cells recover for 6 hours, we can see a restoration of this interaction in cells. Again, arguing that the TPR domain of CHIP is a very labile domain that is sensitive to heat stress. So we wanted to kind of take a look again at using NMR to see if we could see other ways to rescue the structural defects associated with heat stress and with SCA48. So in this olive color here, you can see an NMR spectra of CHIP now bound to the C-terminal peptide that corresponds to the C-terminus of chaperones. And what I'm showing you here is that if we simply take CHIP at 45 degrees Celsius and add an excess of this peptide, we go from seeing this unstructured region to something that really resembles closely to the wild type CHIP. So adding in something that binds to and stabilizes this TPR domain can correct many of the structural defects that are associated with elevated temperature. And so we use this data to hypothesize that the same thing could be true for SCA48. Potentially, if we add in something like this peptide that stabilizes that interaction, we may be able to force this protein to refold. And that's exactly what we saw. Again, in this olive color is wild type CHIP with peptide titrated in. And if we look at the SCA40 mutant, and add in excess peptide, we're able to restore binding to molecular chaperones. And so the reason that we're excited about this data is what this argues is that we should be able to potentially identify small molecules that can do the same thing. So one of the active projects in the lab currently is doing high throughput screening to identify small molecules which stabilize the TPR domain of CHIP, which we think will be useful for treating specific mutants that cause SCA48. So overall, kind of the story that I've told you about today is that CHIP had been really well studied for about 25 years. And the classical thought is that CHIP interacts with molecular chaperones, mediating the ubiquitination of misfolded proteins to target them for degradation. What our studies found is that CHIP actually has many non-canonical functions, one being transcription. So we have a project really focusing on TF2A in the lab. It's very active, looking at how CHIP alters that. But we have a number of other things that came up from our BioID screen that interact a very similar mechanism binding the TPR domain of CHIP. And we have a lot of data that these different processes are also dysregulated due to SCA48 mutations and can be restored by simply restoring the folding of that TPR domain. So we're really excited not only about the potential translational ability of some of these studies, but also think that we've uncovered a number of new cellular pathways that are regulated by CHIP. So our understanding of the basic biology of the protein has really, I think, dramatically changed as well. So we're excited to move forward with these projects over the next few years. We're also really excited to continue studying how this folding and unfolding of the TPR domain occurs, because one of the things that I didn't talk about and has been noted actually through others as well is that the affinity for the proteins that bind to the TPR domain of CHIP is actually quite a bit lower than HSP70. So understanding how they can move in and out of this TPR domain is important, and potentially these structural changes that we're observing may be important for that as well. So with that, I'd just like to thank different people in the lab who have participated in this research. So a lot of this work has been driven by a graduate student who has also been supported by the National Ataxia Foundation, Ron Ming. And she's working with a number of other people, including Adam Kanak, who was a former graduate student who really started this project, and Jamie Scaglione, who collected a number of experiments for this as well. So with that, I'd like to thank the funding agencies, the National Institute of Health, especially the National Ataxia Foundation, which has provided a lot of the seed grants, which have allowed us to start these new projects in the lab, and our collaborators who've helped us with different aspects of this experiment. So with that, I'd be happy to answer any questions that anyone may have. Thank you.
Great. Well, thank you so much, Dr. Scaglione, for that talk. Really, really interesting and cool research. And just a reminder to anyone in the call, you can type in a question, but using the Q&A feature, or if you'd like to ask the question live, just raise your hand and we'll unmute you so you can ask the question. So just a quick question. The data on nuclear localization of CHIP that you showed was quite striking. Is there anything known about the impact of blocking the translocation of CHIPs in the nucleus on known SCA48 phenotypes?
Yes, there's not currently. So that's something that we're following up on. So we can use a number of tricks to put things like nuclear localization signals to make wild type CHIP go to the nucleus, put export signals to keep the mutant from going in. So that's something that we're actively interested in looking at. And one of the other projects in the lab is actually screening for modifiers, which basically control the import and export of CHIP from the nucleus.
Great. And can you speculate on how you think the ubiquitination of TF2 alpha impacts its function?
Yeah, so that's still something that we're working on. Our hypothesis is, and we have some preliminary data to support this, is that it's altering the complexes that TF2A forms. And we see this with some other proteins as well, where CHIP appears to remodel protein complexes. So that's something that we're actively pursuing with the hypothesis that CHIP's necessary for getting TF2A to the proper spots on DNA with TBP to drive transcription.
I'm just having a little trouble unmuting someone, so I apologize. Give me just 1 second. Sorry, Amy, I still can't unmute. Oh, there we go. Kathleen, if you'd like to ask your question, go ahead.
I'm not real good at this screen stuff, so thank you so much. I'm a registered nurse, have been involved with Ataxia services and research since 1977. And my first question after this is absolutely a wonderful presentation. Thank you so much. How would the clinical application of this recognition of folding and unfolding proteins, and with the help that you've been able to identify, do you see yet any projection in the future for an actual therapeutic intervention for individual patients, or is that off in the horizon somewhere?
Yeah, so that's an excellent question. So that's something that we're actively working on. So as I mentioned, we have set up high throughput drug screens. So our goal is to identify small molecules that regulate the stability of this this domain of CHIP. So I think that those potentially could be one way to treat specific mutations that cause SCA48. Because CHIP's more generally neuroprotective, there's always the potential that, therapeutics that alter chip function in some way could potentially treat other diseases as well. But that's something that one part of the lab's actively pursuing, which is the translation of these findings to something that may be more useful in the clinic.
Oh, that's wonderful. Thank you so much.
Thank you.
I do have another question from a spouse and mother of someone with a related disorder. So the first question was, husband showed gait-balanced issues and dementia over a few years ago and confirmed with cerebellar atrophy and diagnosed with ataxia. His TDP repeats are 40 and 42, and he has a STUB1 disorder. Does he have SCA-17 or SCA-48? And how can they figure it out?
Yes, so that's a great question. That's probably something more for someone more clinical than myself. Yeah, I think it's kind of an active area. So not every SCA48 mutation necessarily has the repeat in TBP. It's a very high number. One study found, I think it was 30 of 31 individuals. Another study found like 78%. Somewhere around there. So it's not 100%, but that's an active area of research, I would say.
It's still up for kind of discussion.
Correct. Yeah.
Now I'm going to let Adam and Avery ask the question.
Hey, Matt, really nice talk. Let's see. Can you hear me?
Yes.
Oh, okay, sorry. So one question I had is in your bioID mass spec experiments, you may have mentioned this, but did you see for your CHIP mutants a reduction in the amount of chaperones that came out?
Yes. Yeah, we did. Basically, if we parse out the data, all molecular chaperones that kind of been in the classical EEVD motif, we saw a statistically significant decrease.
Okay. Yeah. There were a lot of proteins that came out in that assay, that mass spec assay. Do you think that there are other proteins other than chaperones that bind the TPR that might be impacted by these mutations?
Yeah, so we currently have 4 projects in the lab looking at different aspects of this. So I didn't talk about it, but proteins that kind of end in the motifs, the amino acids that would bind to the TPMR domain were statistically significantly increased. So we identified roughly 15, I believe, if I'm remembering off the top of my head, different proteins that we predict function the same way.
Okay, well, interesting. Lots of new biology to explore.
Yes, yeah, lots of fun stuff to do moving forward.
All right, thank you.
Thanks, Adam.
Another question from the audience. Given how important STUB1 is for many cellular processes, could you imagine a gene silencing or gene targeting approach being used for SCA-48?
So yes, that's potential. So what I would say is the issue with that probably would be that CHIP is so important that reducing its levels, we know from SCA-16 that seems to be what these mutations do, would be detrimental. So I think, if you had ways to selectively reduce the mutant allele, that could potentially be useful. Or potentially in some of these mutations, which the ones I didn't talk about, they're clearly loss of functions which are caused by truncations, which you probably are missing most of the protein. So a gene delivery system could be useful in some of these cases as well. And so I think that's something that potentially could be looked into as well.
Another question from the chat. What is known about the folding of CHIP in response to other forms of cellular stress, such as ATP depletion or oxidative stress? Is it a similar response to heat shock?
So those are ongoing studies. So what I can tell you is there are studies looking at, the proteins that are immediately oxidized. CHIP's one of the first proteins that gets modified by oxidative stress. But as far as ATP depletion, those types of things, those are all things that, we're definitely thinking about and actively pursuing. So we're curious to see, how this translates into other stressors. And this potentially could be a general mechanism by which CHIP, recognizes cellular stress to alter its complex formation to facilitate different biological processes. That's kind of how we're viewing it.
And there's a couple of questions from the audience about how much in common SCA-48 shares with other SCAs, in this instance, SCA-42, or how close is it to a diagnosis with Parkinson's disease? I know you're a researcher,
Sure.
not a clinician, so feel free to answer as much or as little as you'd like from this question.
Sure. Yeah. So what I can say is that SCA-48, I'm not familiar with SCA-42 specifically clinically, but for SCA-48, there is the typical ataxia, phenotype or symptoms, but there's also a cognitive component, which puts it more closer to, something like Alzheimer's or dementia-based disease as well, that seems to present in a majority of patients. But probably would need someone with more clinical experience to really give you an in-depth answer there.
So another question from the Q&A. What advice do you have for graduate students interested in researching ataxia?
Yeah, and find a good lab, find a place where you think you can do impactful research, and then it's really just about getting in there and doing it. So I think it's a field that needs to grow, and we need to do more to understand these diseases. And I think the biology that we learn from studying things like ataxia or more rare diseases is actually extremely important. So I think that is critical for us having just a broader understanding of what happens in degenerative diseases of the brain.
We have a question from Phyllis, and just a reminder that Dr. Scaglione is a researcher, not a clinician. So let's try to keep our questions related to the research. Go ahead, Phyllis.
Thank you very much. My husband, already has ataxia. The question in between the SCA-48 and SCA-17 was asked by me. It is because we kind of heard a different opinion from the different neurologists already. That's why I asked. And the second thing is that my son is only 4 years old. He's at very high risk, of ataxia, and likely, the SCA-48, because he does have the STUB1 disorder, and his TBP is 35 and 40, and he did show a lot of the developmental delay and motor balance control issue at this moment. I wonder, I'm not, a professional in this area. I wonder, based on your research, how many years do you expect, we'll get, maybe a drug to trial, for example, a new clinical trial, new clinical trial for the SCA-48, and typically how much funding do you think we'll need for this kind of early research? Yeah, thank you.
Yeah, so what I'll say as far as drug discovery goes is that we're on the early stages of it with SCA-48. We've only known about the disease since it was I think December of 2018 was the first clinical paper that was published on it. So there's a lot more work that needs to be done to kind of move things into the clinic. So yeah, so it's an active area of research, in our group, and I know in others as well. So.
Okay, thank you. Do you think let's say the recent AI technologies kind of things can help to accelerate, the drug research? Do they helpful?
So I know a number of labs do use, AI-based approaches for different things. It's not something that we currently use, but anything that can accelerate our progress is worth exploring.
Okay, thank you.
Thank you.
All right. Well, I think that is all the questions that we have today. I want to thank again our speaker, Matt Scaglione, for a wonderful presentation and for some really great work he's done the last couple of years. And then I just want to mention that this webinar series will be a monthly webinar series, and we already have our next talk scheduled for October 27th, which will be given focusing on the cerebellar vulnerability in SCA1. So thanks again, Matt, and I hope you all have a wonderful rest of your day.